from Microbe TV, this is TWiEVO, This Week in Evolution, episode 23, recorded on September 18th, 2017. This episode is brought to you by Blue Apron. Blue Apron is the #1 fresh ingredient and recipe delivery service in the country. See what's on the menu this week and get 3 meals free with your first purchase with free shipping by going to blueapron.com/twee. Hi, everybody. I'm Vincent Racaniello, and you're listening to the podcast on the biology behind what makes us tick. Joining me today from Salt Lake City, Utah, Nels Elde.
Hey, Vincent. Great to be with you again from the iconic Elde Lab Studios.
Iconic, huh?
I don't know. I guess I kind of went for it there.
That's good. I don't know.
Yeah.
How's your weather out there? Nice?
It's beautiful. It feels like summer is slipping away from us, but I'll take it. Yeah. Sunny, cool evenings. Perfect.
You have sun today? No clouds?
Some clouds, but right around 80 Fahrenheit. Good temp.
Nice.
Yeah, exactly.
Yeah, we're all clouded up today. We have some hurricane action offshore, you know?
Oh, right. Yep. What's the latest?
I don't know. I think this is Jose. I'm not sure. Mm-hmm. But, that's something you don't get out there, right?
No, we don't. So actually, we did have a little bit of intrigue earlier this morning. There was a 3.3 magnitude earthquake in Park City, about 30 minutes from Salt Lake. And, it was the building just quivered for about 5 seconds. And I thought it might have just been the heating duct maybe was messed up, starting up for the fall. But actually on Twitter and a few other sources quickly saw that there really was a small tremor out here.
Neat. I felt one here a couple of years ago.
Mm-hmm.
Just a little. I was talking to one of my students, and all of a sudden we felt this shaking. So I went right to Twitter, and immediately people were saying, "Hey, did you feel that?"
That's right. It's interesting how that's the first thing you do in one of these things.
And yeah, it was a little tremor, and it lasted for like a minute.
Uh-huh.
And it's kind of neat, especially if it's not too serious.
Well, that's for sure. So in Hurricane Jose, remind me, so that's the one, it was kind of trailing Irma for a while, and it was just sort of circling around.
Yeah.
Yeah.
It was wandering out at sea mostly,
mm-hmm.
going back and forth, so it didn't hit much. And today, the weekend was pretty nice around here, nice, sunny, and high 70s. But this morning, it's all clouded up and a little bit of rain, but still in the 70s, so it's kind of nice, but it's definitely cloudy.
Yep.
Don't see any sun out there. I don't know what the long range is, but yeah, we're out of summer, kind of slowly slipping away. It's kind of sad because I really like summer, and I think it's the best season. I love the heat. I love being outside.
Yep, you and me both. I guess it's time for our annual end of summer rundown.
Sure.
What did you? Yeah.
Well, I let's see. Last time I saw you was in Madison, right?
Yeah, that's right.
Madison, Wisconsin, American Society for Virology. Although you disappeared after a couple of days.
I did, and one of the things we did, though, a couple of things were on my menu, both a grant deadline, and then I hit a meeting out at the NIH, kind of back to back.
In Bethesda, right?
Yeah, exactly.
Mm-hmm.
And then, but I think the last time I saw you in person at Madison was on Rob Kalita's dock. We were fishing.
That's right.
For sunnies, and came up empty, bluegills. So I got back out a couple more times this summer. Some trips here in my backyard, the High Uintas Wilderness, and on a couple occasions started catching some trout. We had one day where we got almost 20 fish.
Wow.
Yeah. Nice. Another day. Yeah, it was fun. Another day with a really beautiful rainbow trout. So made up for lost time.
After ASV, I was here for quite a bit, but at one point, I think the end of July, I went out to Hamilton, Montana to Rocky Mountain Laboratories. that they were having an arbovirus meeting, and they asked me out to do a podcast, which we did, and had Diane Griffin as our guest. So that was a lot of fun.
Yeah. Cool.
And I brought Dixon de Pommier out with me because he likes to fish out there. He's gone out there many, many years. Avid fisherman. So he took me on a float down to Bitterroot one day. And, I don't fish for trout. And it was a lot of fun. I sat in the back. We had a guide, and we went down on this raft. And Dixon, he must have caught 25 fish. He was just knocking them in. Cool. Mostly rainbows and cutthroats.
Yep.
And that was a lot of fun, I must say that. I just sat there. I actually videoed him fishing and watching him cast back and forth. The guide would say, right over there in that little pool, and he would cast, and bing, the fish would hit. I found it interesting, the whole ecology of fishing. All these little white moths were wandering out of the woods, or these pine forests around there. And Dixon says they get lost, they flutter around, they fall into the water, and the trout eat them. And we could see them. They'd fall in the water, bing. And so they had lures or flies that are mimicking these white moths.
That's right.
It's pretty cool.
Yep.
So that was a lot of fun. But for the rest of the summer, I wrote grants with my scientists here, Amy. We wrote 3. We just put a third one in this week. Oh, wow. But we did submit 2 manuscripts. One of them was my pick a couple of TWiEVOs ago. We put it on bioRxiv. We submitted a second one. We got reviews back. They're pretty good, so it looks like minor modifications. One's an Entero 68 paper. One's a Zika virus paper.
Cool.
So she's been very productive.
That's great.
And that brings us here to September.
Yeah, good summer.
It's a good summer, yeah.
Yeah, yep.
And now back to work, right, Nels?
That's right. And our work for today is some podcasting about a really interesting paper, actually.
A paper that you called spicy.
Yes, guilty as charged. I think this is hits on, it might not be at the surface of it kind of pure evolutionary biology paper, but definitely picks up on some great themes that we're trying to kind of mingle here. Sort of these multidisciplinary papers that have very clear evolutionary implications, or the deeper you get into the study. And I think we'll see that today.
Yep, for sure.
So the paper itself the title is DHX9 suppresses RNA processing defects originating from the Allu invasion of the human genome. And actually, so I had this paper had sort of missed my radar, but you came across it. And so what was it that caught your eye about it, Vincent?
Well, Amy actually sent it to me.
Okay.
I thought it was interesting because we know, as we'll see, Allu elements, we'll talk more about these, but these are basically transposable elements, I should say, that invaded the genome a long time ago, and there are many, many copies of them in the human genome. And we're kind of interested in, retroelements in general on this podcast and many others. They're kind of interesting, how they have invaded the genome and multiplied and evolved. So I thought this would be a cool paper to think in those terms.
Yeah, completely agree. So in the senior author on this paper is a woman named Asifa Akhtar. She has her lab over in the Max Planck in Freiburg, Germany. One of the locations, this is each of the Max Planck institutes have sort of different themes. The theme of this one is immunobiology and epigenetics. Have you run into any of Asifa Akhtar's work before?
I have not, and I took the opportunity to look at her video that you posted. I really liked it. It's cool.
Isn't that great? What a cool sort of, I think, clean explanation of some of her work. And
however, Nels.
Yeah.
First of all, this latestthinking.org is a really cool site. I'd never come across it before.
Yeah, same here.
And secondly, she is explaining the problem in simple terms. She says the DNA in all of our cells is the same, but, some cells become a kidney, some become a brain, an eye. But of course, the DNA in our cells is not all the same, right?
Sure. Yeah.
For the most part, it's similar, but every cell's DNA is different from the next, right? Pretty much. Because of mutation, right? So
absolute somatic mutations.
But I understand why, if you're going to explain it to a lay audience, you want to just that's the basic problem, is that the DNA is basically the same. So how does it code for different things? And that's what her shtick is, and that's cool. But
absolutely, yeah. And so I actually don't know much about her work, but it definitely falls closer to the epigenetics side than immunobiology. Although it will, as we'll see in today's paper, will actually be pulled a little bit into the thinking about some immune-related ideas.
Yes.
But I think my impression is that most of her work, or sort of the foundation or basis of it, is a lot of it's in Drosophila, the fruit fly, and focused on this really cool phenomenon, which is dosage compensation. And this is important not just for flies, but many organisms. And it's this question around the differences in sex chromosomes and how you sort of modulate or deal with the difference in gene expression levels if you have 2 copies of, say, the X chromosome as a female mammal, or 1 copy of the X chromosome as an XY male. Mm-hmm. And so there's really fascinating biology behind this idea of how you deal with the gene dosage on sex chromosomes, including in Drosophila, one of the main places.
By the way, Nels, can you pronounce the first author's name?
Oh, boy. Let's see here. First, I have to bring it up on my screen here. So this is a postdoc in Asifa Akhtar's lab. TWiEVO Akhtar.
Got me.
Probably getting that wrong.
It's Turkish, your name, you think?
That's right. Yep.
I think the first 2 authors shared equally. TWiEVO and Ibrahim Afsar Ilik.
That's right. And I think both are training in Asifa Akhtar's lab. And then there's a couple of other collaborators, all in Freiburg, Germany. Another one at the Max Planck there. And and also the Institute for Informatics at the Albert Ludwigs University, and then finally someone on the in the biology department at the University of Freiburg. So it looked like a local 3-way collaboration between 3 labs, sort of combining biochemistry with some genetics, with some genomics, and then, as we'll see, even some innate immune biology kind of coming in at the end.
Yeah, there's quite a range of technologies used where, in this paper, no one lab is likely to have it. All of them, right?
Yep, exactly. Sort of echoing on this idea of how kind of combining different backgrounds and skills, you can really start to push the biology in new and interesting ways, which I think we saw in this paper.
So this paper, the title has Allu in it, and it also has DHX9. Which do you think in their lab came first?
Yeah, so my suspicion from reading the sort of introductory paragraph is that actually DHX9 sort of was the first thing.
That's what I think too.
Yeah. And then it circled the Alus is in the title.
Yeah.
But it kind of circles back to that. And so I guess maybe why don't we start with DHX9 and then we can circle ourselves back to the biology that this RNA helicase is mediating. And so the paper starts out with this, I think pretty honest, if you will, sort of assessment that here is a protein that is abundant, it's essential, and a little bit is known about its biology, that it's a helicase. This is a protein that binds to an RNA helicase in particular. So it's a protein that binds to RNA, can interact with it, and in fact has the ability to clip or cleave RNA in this case. But other than that, very little was known about sort of what biology DHX9 was sort of conducting, other than being essential.
So what kind of RNA will this interact with? Double-stranded RNA mainly?
Yeah, exactly. And so that will become a very important part of the study moving forward, is that there are sort of sets of different helicases that interact. So helicases could interact with DNA, or they could interact with RNA, both single strand or in this case, double strand RNA.
Okay.
Yeah.
So this is a protein that, as you say, we don't know much about, and it's ripe for some adventurous person to go after, right?
Yeah, I guess so. And I guess it was this lab that did it. And actually, I think maybe at the end we'll return to so DHX9 is a mammalian protein. So and we'll be considering it both in humans and in mouse. But in fact, there is an ortholog in Drosophila, and I think this will maybe at the end we'll turn to that idea and how that might have actually been where the project came from.
And I think DHX means dead box helicase, right?
Correct. Yep. And these proteins, these helicases, are showing up, or at least have come on my radar in the last year or so, as more and more people are pulling them out of screens, especially some involved in immune-related activities. So Sarah Cherry's lab, for example, keeps running into dead box helicases and sort of lifting them up from their genetic screens.
What's a dead box now?
Dead box.
Is that like the Dead Sea or something?
Not quite. It's a motif. I'm trying to somehow tie it to that. No, so the dead box, that's a motif, a set of amino acids, DED or DEXD, or where the X could be anything, that sort of dictates the activity.
Right.
Yep.
Okay.
So, yep. So then, and actually the point you raised, Vincent, I think is an important one. So that the authors state that, there's this sort of collection of double-strand RNA binding helicases. Not a lot known about them. So why not go ahead and take sort of a biochemical approach to ask what other proteins, or actually, sorry, in this case, what RNAs in particular might the dead box helicase DHX9 be interacting with or associating with?
Right.
And so in order to investigate those interactions, they use a method called UV cross-linking in affinity purification, or UV CLAP. And this is
it's not a venereal disease.
Not to the best of my knowledge. But it is sort of a variation on another technique called chromatin IP, or CHIP, where you have basically a protein, in this case for CHIP, which is familiar maybe to some of our listeners. You have a protein that interacts with DNA, an antibody to that protein. You allow the protein to interact with the nucleic acids, and then you use the antibody to basically pull out both protein and DNA. You dissociate the two, and then you sequence everything, and you figure out what DNA that protein is touching. So here, and a variation on that technique, you can find RNA targets. And so in this case, the dead box helicase is allowed to just interact with its regular constituents, and then they're UV cross-linked in order to sort of freeze those interactions, and then an antibody is used to purify out that protein and the RNAs that are cross-linked to it, in order to then get some ideas about what this protein interacts with, sort of during its day job, so to speak.
And they compared human, mouse, and fly, right?
That's right. And so
this is funny now, is the ortholog in flies. It's called maleless.
Yeah, exactly. And actually, so we'll get back to this maybe, but so maleless, this gets back to this idea of dosage compensation, actually. So that protein is interacting, the ortholog in flies is interacting with RNAs that somehow, so these fly proteins or genes are named often after the phenotypes. And so maleless, you can imagine what the phenotype is. If you are missing that dead box helicase, you end up with all females.
It's a great name.
Yeah, I agree. There's so many good ones from Drosophila. But so their main focus to begin with was on the mammalian cases, the human and the mouse. And what they found were all kinds of associations between DHX9 and Alus.
Alus, right.
Yep. And so,
are we going to tell them what an Allu is, or we're going to wait?
Oh, yeah, no, let's do that.
Allu is a restriction enzyme originally, ALU1. I don't know if you use restriction enzymes still, Nels, in your lab. Do you?
Of course, all the time.
Because back when I started, that was when recombinant DNA began, and we loved having all these restriction enzymes. ALU1 was purified from Arthrobacter luteus, so they used to name them from the bacteria that they were purified from.
Yep.
And of course, my favorite was Bagel 1, BGL1. But not everyone pronounced it bagel. Only people from New York City. What else would
pronounce that one?
Would you pronounce it with? But Allu 1 was a 4-base recognition sequence, AGCT, and it would cut between the G and the C. Mm-hmm. So Allu elements, and Nels will tell us in a moment what they are characterized originally. We didn't know they were transposable elements. We just knew that there are lots of Allu sequences in the genome, say, of a human.
Yeah.
You know, if you cut human DNA with Allu, you would cut it, and basically you get about 5 kb pieces. So the idea was there was a Allu element every 5 kb. And so back in the 70s and 80s, when we didn't have sequences of many genes, we could use Allus to identify genes.
Yep.
So we actually did that in my lab when we cloned the poliovirus receptor. What we had done was to take human DNA and introduce it into mouse cells. Mm-hmm. And we found that mouse cells could be infected because we thought they were making a receptor. And we didn't have any way of getting the human DNA back out. We had no probes, right? Mm-hmm. So we used an Allu probe.
Cool.
And just used that as a hybridization probe to pull out clones. And eventually we got the receptor that way. But this is lost now. You don't have to do this anymore, but you can just sequence the whole thing. But
that's right.
That's what we used Allu for. And now, I don't know how many years later, it was figured out that they were transposable elements, right?
Yeah, that's right. And so it wasn't long after that became known. And so the fact that 4 base pair recognition sequence you mentioned, that AGCT, that palindrome, that's found pretty liberally in these retrotransposons. And as you said, these things have then sort of propagated throughout the human genome. In fact, I think the estimate is that about 10% of the human genome is actually made up of these elements. And that's why between that very, kind of simple 4-base recognition and the prevalence of these things, that you just, those restriction enzymes just chop the genomes into tiny pieces because they're so easy to find those sequences.
Yeah. Yep. And this Allu element is basically a copy of a small cytoplasmic RNA, right?
Yeah. So there's this really interesting sort of deep evolutionary history for these things, that it's these cytoplasmic 7S LRNAs, which are part of the so-called signal recognition peptide. This is involved in it's been sort of developed somehow in eukaryotic cell biology to be part of protein secretion. If you have these SRP signals, these are proteins that will be trafficked through the endoplasmic reticulum, through the Golgi, as part of the secretory cargo. But through some ancient associations, part of that RNA sequence was then sort of repurposed or kind of recombined to become what are these transposable elements, these selfish genes, if you will. And in particular, the Allus are part of, they're a primate-specific example of what are known as SINES, or SINES, short interspersed nuclear elements. These turn out to be non-autonomous, so they can't selfishly copy and paste on their own, but instead are sort of given a helping hand by another set of retrotransposons called lines, or the long interspersed nuclear elements. And in fact, the lines have been super successful in our genomes, and nearly a quarter of the genome is devoted to line elements. And so in a sense, you have then this teamwork where the line open reading frames encode proteins that associate with the Allu RNA, and then allow these things to become sort of parasitic hitchhikers in the genome.
Do lines encode reverse transcriptase, Nels?
They do. Yep, that's correct. Yep.
And they provide that to copy the sign elements, and
exactly.
And that's how they multiply and proliferate, right?
That's exactly right. Yep. And so, as you can imagine, as these things are, propagating, just kind of jumping, copying and pasting through genomes, this could be, from an evolutionary standpoint, sort of like throwing around genetic hand grenades, where you have the sequences reverse transcribed into new genomic locations. And as we'll discuss in some detail, this could have a big impact on gene regulation. If the Allu element lands in the introns or in and around the exons of a gene, this could really change the way the gene is transcribed, the level of transcription, whether the gene is expressed or not. It could influence how the genes are spliced. So you might get one exon included or excluded, depending on how the RNA from the Allu element is being transcribed in that area, and even in some cases interacting back in that genomic location. Yep.
Now, what is it about Allu that made it so successful? Because, in theory, any sequence could be turned into a sign or a line, but something about it now, Nels, something about Allu, right?
yeah, right. Yeah, it's a good question. So, and we talked a little bit about kind of along these lines in a previous TWiEVO. I can't remember offhand, but there are these micro or eukaryotic cells that have another whole class of transposable elements called introners.
Yeah.
And remember when we were kind of talking about possibly the origins of introns?
Yes, that's right.
Sequences between, yep. And, for whatever reason that these sequences seem to, show up in and around introns, in and around open reading frames as they're propagating. There's just, something, and as with sort of all mutations, it's sort of this double-edged sword where, you can throw these hand grenades that could be explosive in the sense that it could really change how a gene is regulated, or it could be beneficial in the sense that maybe this allows something to be turned on in a new way or regulated in a new way. Or another major class of just neutral, that these things just sort of hop around and it doesn't have a functional consequence. And so between all of those, there's some almost evolutionary calculation that's been weighed and built on for millions of years that sort of these Allu elements have sort of threaded the needle in the sense that they haven't been too destructive, and at the same and are also really able to copy and paste. Yeah.
All right. Sorry, I disrupted you from talking about the UV clap results. What did they get there?
Oh yeah, no, not at all. So what they found basically was that as they were cataloging the RNA regions in the RNA that the protein bound and where that RNA sits in the genome, that something like 60% of these sequences actually lined up with Allu elements, and that's and they have a panel in Figure 1 with 5 or 6 other RNA binding proteins that are not this helicase DHX9, and on average those sort of controls would be associated with or bound to 3 to 10% of Allu. So quite an enrichment. I would say though at the same time, because as we've kind of discussed with the Allu1 restriction enzyme, and just because Allus are so prevalent, you always want to be a little bit careful when you pull that out. As an experimentalist, your first thought might be, we're just getting kind of non-specific binding, or is this just background?
Yeah.
Because there are just so many of them that you can kind of imagine being fooled. Or at the same time, you could also, just sort of psychologically as a researcher, you might get these Allus and just think, oh, well, that's not interesting. There are millions of these things, and how am I going to find some really cool biology if it just associates with, millions of these things? There's what's really going on here. And so, it's possible that I can imagine even myself, like if I got that result, being a little bit disheartened, and sort of just thinking, well, are there any rare RNAs that might be doing interesting biology, instead of like sort of stepping back and thinking that, well, this is the biology. But I think to the credit of the authors, they did follow this up and I think did some nice controls actually to first of all show that enrichment of 60% binding versus 3 to 10% might be real. And in fact, immediately they use another cross-linking method, this flash cross-linking.
Another acronym. Right?
Yeah, I can't remember. Don't ask me do you know what that one is?
Yeah, the flash is, I got it right here. Fast ligation of RNA after some sort of affinity purification for high throughput sequencing. See, that's the whole thing. F is fast, L is ligation, A is affinity, and S high throughput sequencing, I guess. But it's a funny they after some sort of affinity purification, little humor there, right?
Yeah, exactly. So kind of a complementary way, right, of seeing whether they got the same result. And the answer actually was yes.
Yep.
And so, and then actually immediately there's sort of a comparative approach that you can take. So in mouse, they have a very clear ortholog of DHX9, the mouse version of the gene. And yet there are no Allus in mouse genomes. However, there are sign elements, these short interspersed nuclear elements. And the mass, it's sort of a parallel evolution in a massive family of so-called B1 elements. And so different TEs, but very similar sort of histories, actually. I think they both emerged from those 7SL RNAs, actually, and they're kind of went their separate ways as the lineages split off. And in fact, they see a very similar result if they use the mouse DHX9 for cross-linking in mouse cells.
In fact, the lack of Allu in mice is what allowed us to identify the polio receptor using a human Allu probe.
Oh, cool.
Otherwise, it would react with mouse DNA, and we wouldn't be able to distinguish it,
Yeah, very cool. Yep. So you took advantage of that.
Yeah.
Yep.
And back then, we used to do southern blots. So if you took total human and total mouse DNA and digested it, ran it on a gel, and incubated it with a P32 labeled Allu probe. So it's about 200 or 300 base pair piece of DNA, which you can cut out of the genome with Allu and clone it, make a probe out of it, a hybridization probe. It will react with human DNA. You'll get a schmear because there's a lot of Allu sequence, and the mouse is clean. So that's the beauty of that. Yeah.
Yep. And here the beauty of it is that you can kind of build these parallel stories, basically. And I think, again, if you're sort of still kind of worried in the back of your mind, is this association with the Allus, is that specific? Is there something real going on here with the biology? Or is it just this kind of biological noise? Basically, the fact that you see it in two independent cases starts to make you think maybe there is something going on here. And then the authors kind of take the next step from there, which is there's millions of these elements, of these signs. And so is there anything sort of specific about them that might also point to some specificity in the biology? And so here you could take, all these increasingly powerful genome-wide approaches to sort of scan through and ask, are there any sort of features that emerge that are specific to the RNAs that might be interacting with DHX9? And so what they found actually by doing this was that if you look at the spacing of the Allu elements, there's a real difference between the ones that were associated with DHX9 and the ones that weren't. And so what they are sort of imagining is that given the fact actually that these RNAs can sort of self-associate with each other, they form this double-strand RNA, which, as you pointed out, this is a double-strand RNA binding protein. It looked like this was associating with Allus that were in closer proximity to each other, that when transcribed could actually form double-strand RNA. And so that was seeing that sort of genome-wide pattern, knowing that the motif or the domains were double-strand RNA binding domains. That actually made sense for that to be enriched. And so that was sort of probably enough to say, yeah, we were probably onto something real here with this sort of big association with all of these transposable elements and families.
Right. Yeah, that's a nice thing that you can do if you're thinking broadly, right? To say, let's compare the different species and see if we come up with something. And it's a good bit of advice. Always think broadly if you can go into another species and get some more information, right?
Yep. And I also think, sort of combining the biochemistry, which are all these pull downs, very technical sort of wet bench approach with then some collaborators or, as folks are kind of cross-training in labs as well, to then think about how do we take these results and then sort of put it in the context of the whole genome. And that takes a little bit of, some collaboration with sort of genome scientists, if you will. Yep.
So this gave them some courage now to go ahead and look at the effects of removing DH9 from cells, right?
Yeah, correct. And so that's sort of, I think the next thing that a geneticist or functional biologist might turn to is to say, okay, well, let's disrupt this protein and then ask what are the phenotypes and outcomes. And in fact, this had been done previously. It was not much was known about this protein, but it was known to be essential. And so, but because they had these clues that sort of tied it to the double-strand RNAs that might emerge from the transcription of these Allus, then they could sort of hone their vision to ask, okay, well, what are some phenotypes that we might predict would come out of depleting these things? And so one of the first things they looked at were for the presence or absence of these so-called circular RNAs. And so do you want to, that's actually, these guys these circular RNAs have just very recently sort of come on my radar. Is that something that you want to describe?
I'll be happy to. Most of us think of RNAs as linear molecules, right? Whether it's mRNAs or ribosomal tRNAs. I mean, they can have a lot of secondary structure, but they always have two ends, a 5' and a 3'. But not too long ago, it was found that, in fact, there are circular RNAs in eukaryotic cells. They don't have an end, and they're kind of enigmatic. There are lots of them in cells. They were originally thought to derive from introns, but now we know they can be derived from coding sequences as well. Mm-hmm. And I know Peter Sarno, who I've known for many years, as a polio guy, and he's also interested in RNAs, very interested in these. He said they can be storage areas for shorter RNAs, like interfering RNAs.
Yeah, isn't that cool?
That they're just like hung up. Think of it as the circular RNA as a hanger, and you're hanging up some other RNAs on it that you might want to use it sometime. It's so neat.
Yeah. Yeah. No, I love it. Yep. These ideas are emerging of sort of like sponges.
Yes.
Like tons of these circles might get made, and then they could act as hangers or sponges in this case to not only sort of supply RNAs, but to sort of suck them up and kind of keep them sequestered into different places in the cell. Yeah.
So Nels, why did they look at circular RNAs? Why was this the next thing?
Yeah. Well, so this is, as you said, how are these things formed? Because the sort of canonical linear RNA, which is well known, and part of the way that these what's emerging is that Allus can have a hand in facilitating this circular RNA formation. And because my reading of it was because they were, obviously implicating these specific Allu or this massive set of Allus, that this could be sort of part of that biology actually that that DHX9 could be mediating. And so, one of the ways that those circular RNAs were sort of first, I think, kind of brought forward was to do RNA sequencing, but to do a trick. And that trick was to first treat the cells with a RNase, a protein that will cut RNA, but specifically will cut only linear RNAs and will spare basically circular ones. And all of a sudden, as you do the RNA-seq and look at the data sets, sort of emerging from these massive data sets are all of these circular RNAs, right?
So they used exonucleases, right?
Yeah, that's right. I think RNase R is one example of that, exonuclease that specifically cuts it. Yep. Okay. And so what the authors did here was just sort of repeat that sort of sequencing, that RNA-seq approach, but to do it in cells that were either, in essence, plus or minus DHX9. And what they saw was when they depleted the cells by the silencing of DHX9, that the number of circular RNAs went from something like 25,000 up to above 50,000. And so that gave them this idea that DHX9 is actually involved in this biology of, so that it's actually holding down the amount of circular RNAs that form.
Yeah, it's very cool.
Mm-hmm.
Which ends there pretty much. They don't take that any further, but you can imagine that you could do some other things that would be interesting here.
Yeah, correct. And so they're starting to kind of build a case that in the absence of DHX9, that your RNAs will kind of do these crazy things, that you have all of these Allus that encode the potential to make new sort of strange RNA forms or associate with RNA in different ways.
Right.
And that this might be sort of holding it in check. And so another example of that, or major example of that, is also sort of involves these Allus that are found as inverted repeats in the 3' UTRs, or untranslated regions, the end of a gene, basically.
Mm-hmm.
And so the idea is, again, that if these Allus are just hopping around and transcribing kind of out of place or out of time, that this will influence actually how genes are expressed or how they're transcribed. And so, again, just using that simple knockdown of DHX9, they find that by taking it away, all of a sudden, all of this translation is de-repressed, or it's unleashed, if you will. And so you have, again, this building story that the DHX9 is sort of important for keeping the whole sort of a lot of transcription to execute in a sort of a behaved or domesticated manner. And if you will, things get kind of wild if in the absence of DHX9 not sort of supervising some of the activity of these Allu elements.
It's amazing. It makes you think of the scenario where these Allus started to proliferate, and they were lethal in some cases, and the survivors had maybe DH9 that was slightly altered, and they survived.
Yeah, isn't that cool? Like, we'll kind of revisit that idea and kind of the evolutionary standpoint, but this sort of balancing act between Allus existing or being sort of harnessed or somehow domesticated in some way. It's actually a really fascinating thing to think about. So, and actually, one little twist on that experiment, which I thought was kind of cool, is they also, so DHX9 is an enzyme; it's a helicase,
right? And
so they could sort of drill down a little bit farther and test. So they took a mutant, they could knock down DHX9, but then put back either a wild type or a mutant that doesn't have that enzymatic activity. And in fact, putting back that helicase mutant didn't rescue anything. So the transcription was still de-repressed. And so that argues that it's actually, it's not just associating with the transcripts, it's actually processing them or chopping them, cutting them in a way that is necessary for it to sort of keep an eye on transcriptional regulation.
Yeah, nice experiment. I like that.
Yeah, I agree. Yep. And then sort of relating to that, so you have the formation of these, or the lack of formation of these, sort of the repression of these circular RNAs. You have the repression of translation for some of the transcripts. And then they just did another kind of simple analysis, kind of in the same vein, which was to ask, are there genes in the absence of DHX9 that are upregulated? That is, are they expressed at a higher level? And so here you can just do, again, RNA-seq in the so genome-wide RNA sequencing of cells, either wild type or knockdown, and then ask, are there genes, and which of them are upregulated? And in fact, they found thousands of genes to be upregulated, and went in and sort of looked at a few specific examples. And I actually like this as well, is that we saw this already in the paper, but this comes up a couple times where they take both the genome-wide view, but then actually pick a specific target and sort of drill in to it. And so they mentioned a gene called CCL25. And this is a gene that codes for a cytokine protein. So it's upregulated as part of sort of an inflammatory response. And what they found was that in the absence of DHX9, that this gene goes up. And so you have now and very significantly goes up. And so you have now kind of DHX9 potentially working on both sides of the sort of gene regulation ledger, that you can have cases where genes are there's going to be more of it or less of it in the absence of DHX9. And so again, this idea that these Allus are just sort of going in and sort of like as hand grenades and sort of changing your regulation and then having a way to cope with that could actually be really useful. Mm. And so here they show, in order to kind of illustrate this, they show these what are called sashimi plots. Have you seen these before?
Yeah, we have. We had a TWiEVO on them, and we actually used that as part of the title.
That's great. Actually, this could be a good time for a Blue Apron.
It could be, yeah.
Ad, yeah, with the sashimi connection there.
You bet.
But this is so. The idea is that how do you visualize basically RNA transcription, especially for these genes that it might be multiple exons. There might be alternate splicing where you mix and match some exons as introns are spliced out or not. And so these sashimi plots are basically a way of visualizing that, where you take all of the exons and then you take your so-called read coverage of the RNA sequence. That every time you pull out an example of a RNA, in this case, that maps to that exon, you start piling it up. And I guess to the authors of the creators, the inventors of this way of visualizing transcripts, it looks a little bit like a plate of sashimi, of sushi, like a little piece of salmon or something. Especially, I guess, if you color your reads orange, then it has that, that sort of fishy appearance, sitting on a plate, and then with little lines that connect exon to exon, that indicate basically how splicing is happening. And so for that gene CCL25, the sashimi plot in the DHX depleted cells is really interesting because not only is it influencing the splicing, but also the transcriptional termination. Mm-hmm. And this is what's really leading to that upregulation of the gene in the absence of DHX9. And what they have done is linked that to the presence of these Allus that are sitting there.
Neat.
Yep.
Well, since you prompted me for a little Blue Apron with sashimi, let me do that now. Tell you about the sponsor of this episode, Blue Apron, the #1 fresh ingredient and recipe delivery service in the country. Their mission is to make incredible home cooking accessible to everyone while supporting a more sustainable food system, setting high standards for ingredients, and building a community of home chefs. For less than $10 per person per meal, they'll deliver seasonal recipes along with pre-portioned ingredients to make a delicious home-cooked meal in 40 minutes or less. And you can customize your recipes every week based on your dietary preferences. Here are some of this month's recipes. Summer vegetable and egg panini with Calabrian chili mayonnaise and caprese salad. Garlic butter shrimp and corn with green bean salad and roasted purple tomatoes. And skillet vegetable chili with cornmeal and cheddar drop biscuits. They have amazing recipes that don't repeat in any one year. You can pick what you want, or you can let them surprise you. No weekly commitment. You get deliveries when you want them. By shipping exactly what you need for each recipe, they're reducing food waste, and their freshness guarantee promises that every ingredient in your delivery arrives ready to cook or they'll make it right. I like Blue Apron so much. If you're not a good cook or you don't like cooking, you can learn because they give you this lovely recipe and everything you need measured out, and you just put it together as they say, and it works. And so if you've always wanted to cook but didn't have the courage, or you don't like shopping like me, check this out. If you want to do it with your kids or your friends, it's kind of a neat thing to stand around the counter and cook, and
yeah,
especially with kids, you can say, hey, let's try this and get them into cooking. This is a good thing to be into, is cooking. Mm-hmm. So, Nels, have you ever tried this?
Well, I'm happy to report, Vincent, I have my first order from Blue Apron coming on Saturday.
Nice.
So I'll be in the game and will report back next episode and let you know what happens.
Love to hear what happens at the Elde kitchen there.
That's right. Elde Kitchen Studios
Wonderful.
will be up and running this weekend with some Blue Apron menu items.
Check out this week's menu. Get 3 meals free with your first purchase with free shipping. Go to blueapron.com/TWiE, TWiE. That's blueapron.com/TWiE. You'll love how good it feels and tastes to create incredible home-cooked meals with Blue Apron. So don't wait. Blue Apron's a better way to cook. All right, back to sashimi.
That's right. Or actually, I think we can transition from sashimi to Blue Apron to grapes.
Grapes, that's right. A little dessert.
That's right. So after I started doing that analysis from genome down to single genes to the sashimi plots of alternate transcripts, the authors then transitioned to say, well, okay, we've kind of thought about the species of RNA that might be influenced by the presence or absence of DHX9. The transcripts themselves then sort of form and potentially function. And then the next major category of phenotypes they looked at was, do the cells have morphological issues, or can you just look at them and say there's something wrong here? And so that's where they described, so they've just knocked down DHX9 and then sort of just let the cells divide and ask, what do they look like? And they described the morphology as grape-like. And this is actually not a term they came up with, but has been observed before. And basically what's happening is the cells, as they're dividing, are failing to undergo sort of a proper cytokinesis. That as the nuclei divide, they're not going sort of one daughter nucleus to a daughter cell, the mother nucleus staying in the mother cell. But in fact, the cells are just starting to pack full of these micronuclei that are all sort of strung together and take on this sort of a bunch of grape-like appearance. And so I think it was pretty clever. So what they did is they, having seen that phenotype, they then went and leaned on some other studies that also described these grape phenotypes and attributed it to certain genes. And then they asked, are there any genes that might be associated with Alus that could actually be linked to that very striking nuclear morphology? And so they, again, drill down to a single gene. This is one called SPC24. And the protein is part of a tether that links together microtubules and chromosomes through the process of cell division. And if this goes off the rails, then you end up with the grape phenotype. And what was really cool, and they show this, I think it's in figure 3, panel A, is that if you just look at that, the exons and the introns of that gene, it's chocked full of Allu elements from end to end.
Yep.
So again, that makes a really kind of cool connection to this idea that, well, maybe, when DHX9 is gone, you're sort of, chaperone's maybe not the right word. Let's say shepherd of these Alus in their RNA. Then perhaps that's causing trouble with SPC24, which you need to properly tether the chromosomes to microtubules to go through cytokinesis. Yeah. And so they make that connection, although they didn't follow it all the way through. So they made what they called a Allu bypass allele. And basically what they meant by that is they just genetically remove those Alus and sort of reassemble the gene without the sort of potential burden of those transposable elements. And what they do show is when they do that, that even in the absence of DHX9, that there is more protein, the SBC24. So the consequence of depleting DHX9 in sort of a normal condition is that the amount of SBC24 protein is down, which is consistent with then having the problems of cytokinesis. But and that if you make the bypass allele, that the protein is back up. But they didn't go on to actually show us whether or not that alleviates that grape-like phenotype. Yeah. So my suspicion
it didn't,
is
right?
that it didn't, right? but that there's, you can kind of wave your hands and say, oh yeah, well, that's one of the players.
Yeah.
And perhaps there are several others that, because it's such a kind of potentially a global effect.
Yeah. It's probably a multi-gene phenotype, right?
Yeah, I think that would be my intuition on that.
Or they're saving it for another paper. Who knows?
Yeah, could be. Yep. But still, again, kind of cool, right? Going from that sort of morphological observation all the way to sort of a molecular prediction on what's kind of underlying that. And then what, at this point in the paper, what could we use but some more biochemistry?
And another acronym, right?
That's right. So this one's SILAC. Do you know what that one is?
Stable isotope labeling with amino acids in cell culture.
Are you familiar with this?
I've never heard of it, but I mean, we used to do this. We used to label with amino acids all the time. I don't know what the SILAC does, and how you would use it to purify things, right?
Yeah. So my impression of this technique, I haven't done it either, but so it's
there's a website called silac.org. Oh my gosh.
Oh yeah. Yeah. I think it's in pretty robust use these days. So it's basically a mass spec strategy. So what you do is you have your, so you're interested basically in whether your protein, so up until now, we've been thinking about how DHX9, the protein, interacts with RNA. And so now they're transitioning to ask, what other proteins does the protein interact with? Yeah. And so you use, the mass spectrometers turns out to be souped up in ways that are really good at doing these sort of proteomics approaches where you can figure out what protein is touching what protein. And so to have some a way of getting specificity or of enriching for certain targets, my understanding of the SILAC technique is that you basically take your cell culture, you have sort of two dishes in the most simplest form. One of them unlabeled, the other is treated with heavy isotopes or sort of a related modification that's taken up or modifies some of the amino acids just in one of the dishes. And then what you can do is, figure out a tag or a way of pulling down the proteins specifically with DHX9, and then just mix everything together. Yep. For the mass spec, and then you can get a ratio of the heavy proteins, which should be basically specifically interacting to the light proteins, which would just be sort of the background of just kind of random collisions or sort of noise in the system. And so from there, they find, I think, something on the order of 80 proteins that are at least tenfold enriched. Mm-hmm. That is 10 times as many show that heavy isotope labeling relative to the to the control plates. And among them, excuse me, one of them that they focus on is ADAR. Another acronym here. We're back in the alphabet soup. What's this one, Vincent?
Adenosine deaminase for RNA, I think. Is that right?
Yeah, I think that's right. Yep. And so here's another, like one of the reasons they pulled this out or lifted this one up for the paper or to continue studying is that it's another double-strand RNA binding protein. Yeah. And so what does ADAR do? Do you remember?
I believe it binds to double-stranded RNA and converts the Gs, the guanines, to inosines, right?
Yeah, exactly.
And that will basically break up the double helix, the double strand, right?
Yep. Yeah. And so ADAR in some forms is actually well known to be kind of important immune defense. Mm-hmm. So if those double strand RNAs that it's recognizing, for example, are from a virus, one way of combating these things is to hypermutate them to make that substitution the G to I. Right. And this can be a sort of first line of defense to mess up those virus transcripts so that they can't go on to be translated. And so here's where now we're making that sort of first direct connection between sort of this epigenetics view of DHX9 as sort of a shepherd of the Allus to interacting with ADAR, part of the innate immune system. Mm-hmm. And so in particular, it's kind of interesting because turns out there are sort of different flavors of ADARs. Ones that are turned on specifically during virus infection, so-called interferon-inducible
Right.
Versions, and then ones that are around all the time. So it turns out ADAR is actually sort of have an important role, even kind of in housekeeping functions, where you're doing G to I substitutions for some biology. But in particular, it was that interferon induced, that immune flavor that was interacting with DHX9, and again, right, there's that you can in this case, both of these are double-strand RNA binders, and so they go kind of go that extra effort. They take one more step, and so they treat the proteins within RNAs and find that most of those 80 proteins it interacts with, actually it's through an RNA bridge that the two proteins are associating through RNA, as judged by the fact that if you cut RNA, then the proteins are no longer associated with each other. And that was the case for this interferon-induced ADAR.
That's remarkable. A remarkable finding that most of them are RNA binding proteins.
Yeah, exactly.
Yeah.
Yeah. So then, so now they have this sort of connection to this other double-strand RNA binding, one that's involved in immunity. And so they do still one more experiment, which is to ask what happens to double-strand RNA if that's depleted in cells. And what they found is, so to measure that, they took an antibody that it's sort of a non-specific antibody that binds to double-strand RNA, which takes on sort of a specific epitope in the cells, and show that if you deplete DHX9, all of a sudden all of these sort of speckles emerge in the cytoplasm. And those speckles are immunoreactivity to the antibody. So what this hints at is that, in fact, you have, with DHX9 missing, you sort of unleash all kinds of double-strand RNAs that otherwise are kind of under the control of DHX9. And so to me, this is really interesting because, so these speckles, we use this antibody in my lab sometimes to look at vaccinia virus, which also forms double-strand RNA in the context of infection. As the virus transcripts are being produced sort of in both directions, they'll overlap, and this becomes sort of blood in the water for the innate immune system to mount a response. And so here, in the absence of DHX9, even and in the absence of any virus, those double-strand RNAs sort of show up. And so I actually thought this was really cool because what this could mean is that without DHX9, the cell might think that it's actually infected, that there's all of the cytoplasmic double-strand RNA that it needs to set off an alarm.
Right.
And so I would have been tempted. I mean, this paper, they did a lot of experiments, don't get me wrong.
Yeah, they did.
A lot of work that went into this for sure. But I would have been tempted to actually ask, is the cell, in the absence of, or in the knockdown of DHX9, is the cell actually sort of fooled into mounting an immune response? And if it was, that could actually imply a really cool role in sort of keeping the cytoplasm free of double-strand RNA, so that's when you are infected. So you don't have this, you sort of keep your alarm set so that you're ready when you actually are infected to mount a response. And so you might actually depend on DHX9 not to just sort of always be in this panic mode, in a sense, if you're a cell.
So do you think these double-stranded RNAs that are appearing in the absence of DHX9, you think these are Allu containing, for example, forming structures from inverted repeats?
I think that's exactly that would be completely consistent with their results up till then, since they find, in particular, that from the very first experiment, that the DHX9 is associating with these Allus that are known to form the double-strand RNAs. So I think you're exactly right, Vincent.
So they could purify those and sequence them.
Yeah, exactly.
That would be worth doing, right?
Agree. Yep. Yeah, and that might give some nice clues about how DHX9 is sort of acting as this chaperone or shepherd of these Allus, in a sense.
This is really a remarkable paper, because there's so much work, as you said, but the findings are really provocative.
I agree. Yep. And so they sort of conclude the study by saying that the elevated levels of DHX9 could act as a preemptive measure against viral invasions, which could increase the tolerance of the host genome for a higher number of sign insertions, these are the Allus, over the course of evolution, which may enhance sign line mediated genomic or transcriptomic innovation in evolutionarily complex organisms. So I think that's a really cool idea, and it gets to, you raised this point earlier, which is basically and we've been kind of talking about it or hinting at it all the way through, is sort of the double-edged sword of having selfish genetic elements propagating through a genome. And on the one hand, these things can be an absolute mess. They can throw a hand grenade into your genetics, change the regulation of important genes that might be involved in something like cell division, cytokinesis, those grape-like phenotypes.
Yeah.
While if you got rid of them completely, you also might suffer sort of on the other side of the other edge of the sword, which is these can be really creative genetic entities. This has come up in a lot of our TWiEVO episodes so far, where basically a transposable element hops and all of a sudden you turn up the expression of a gene that can allow yeast to become clumping together from TWiEVO 21, or remember all the way back to the peppered moths and how that involved a transposable element. There's sort of story after story of how these signs or lines or related transposable elements can change transcriptional regulation in sort of rare but positive ways that evolution could act on.
I think it would be really interesting if they took some of these DHX9 null cells and infected them with some viruses that will produce double-stranded RNA during their replication cycles and see the effect. I mean, it could make infection worse, or there are other proteins in cells that deal with double-stranded RNA, so you might not see a difference. But because their idea is that these have some role in viral invasions in addition to sign invasions, so that would be interesting to test.
Yeah, I agree. And actually, the data they have so far, which is just that association with the interferon-inducible ADAR, my interpretation is a little bit different from theirs, which is that perhaps DHX9 isn't blocking viral invasion, but in fact, it's as I was saying a few minutes ago, it's just sort of keeping your signal kind of in reserve for cases when you really do want to sound the alarm that you're infected with a virus. And so actually that could be very adaptive in the sense that you don't want to turn on your immune system unless there's really viral double-strand RNA. And so if DHX9 is keeping the sort of host RNA quiescent or quiet, that could be what's going on with that. But I agree, Vince, it would be really fun to see if along the lines they're suggesting this preemptive measure against viral invasions. That's another, I think, viable hypothesis at this point. Yeah. To be tested. Yep.
I mean, there are other systems for dealing with RNAs. So, PKR and MDA5 and RIG-I. So it may not need DHX9, but it would be interesting to do the experiment.
Totally agree. Yep. Yeah. And PKR, that's one of my favorites.
Yeah.
We studied starting as a postdoc, I studied its evolution,
that's right,
which turns out to be pretty spicy as well. Yeah.
Lovely paper. Nice.
Yeah, very cool. I'm also hoping maybe in a future chapter, we might hear more about that Drosophila version, that malus.
Yeah, that'd be cool.
Yeah. I wonder if it's doing some, sort of the same activity in terms of binding double-strand RNA, but perhaps not shepherding Allus, but in this case, influencing the dose of genes more directly. Who knows how much overlap there is. Yeah. Well,
Nels, I have a podcast looming at 5:30, so let's get on to some.
Oh yeah, back-to-back podcasts.
Not good scheduling, but.
Yeah.
What do you have for us today, Nels?
Yeah, so I'll charge right ahead here. So my pick of the week, Vincent, is a short video as part of the series from Joe Pelka. Have you run into him, the NPR science reporter?
Not physically, but I know of him. Yeah.
Okay. Yeah. Don't run into him physically. Nice guy, but
I guess you have. Have you run into him?
I have. I had a meeting. Many years ago. It was really fun to meet him. And so what he's been up to somewhat recently is this. He has this series that he calls Joe's Big Idea, and the tagline for it is exploring how ideas become innovations and inventions. Cool. Yeah, it's really neat. So I've got a link to his website. And currently at the sort of top of the list, the story that they did was called Saving Hellbender Salamanders. And so one of the Joe's Big Idea correspondents, this is a woman called Maddie Sophia. There's a great just 3-minute video, and this will be my pick of the week. I've got the YouTube link here of reintroducing the salamander, the hellbender salamander, to southern creeks in southern Ohio. And so what I like about this video is two things. First of all, the hellbender salamander is just an incredibly interesting creature, great amphibian. But then also from sort of a science reporting standpoint, I think it's a great video, really kind of punchy and interesting. And I would definitely recommend it's only 3 minutes. Just watch to the very end. There's a good sort of science joke right at the end there.
Yeah, it was fun. I liked it.
Yeah. How about you, Vincent?
It's good stuff. Well, I saw that you had gone fishing this summer, so I have a pick, which is not just for you, but anyone who likes the fish. It's a website that I helped Dixon Despommier put together. It's called The Living River, Stream Ecology for Trout Anglers.
Really beautiful.
It's all about ecology, and he's got lots of photos, river types, like the freestone, the feeder stream, the limestone stream, Spring Creek, how energy flow works in these things. He's got a lot of pictures because he's done a lot of fishing himself, glossary, appendices, salmonids of the world, etc. Wow. Lots of links, and a trout shop where you can buy his 2 books one of them is about fishing. It's called Waist Deep in Water. It's a little memento of his for 99 cents. It's full of pictures and words about his fishing. All in all, just a beautiful site. And I thought you might like it, Nels, and other people as well.
Yeah, thank you. I like it a lot. And actually a nice overlap with the hellbender. So you don't want to catch a hellbender salamander on your fly rod, but the stream ecology is super important for this species as well. And so part of the reintroduction efforts for the hellbender is that a lot of the streams, as they become silted, or the siltation, just the accumulation of small gravel or sand, that this actually plugs up the rocks that the baby hellbenders need to hide out and grow. And so, this idea of stream ecology, not only for trout anglers, but also for sort of salamander enthusiasts as well.
That's very cool. And also the site you picked up before, latestthinking.org, we have a video of Asifa Akhtar, and it's really, I like their approach. They have the person sitting on one side of the video, and then they have some illustrations on the other side. It's kind of neat.
It's really good.
So hers is there. Hers is very good. And then lots of other people as well.
Yep. Another good resource.
Good stuff. All right. That is TWiEVO 23. We're moving towards middle age here.
Coming up on that 2-year mark. 2-year anniversary.
This month is my son's 23rd birthday.
Oh, wow. Very cool.
You can find it at Apple Podcasts, microbe.tv/TWiEVO. Of course, if you listen to podcasts on your phone or your tablet, use an app, you can subscribe to TWiEVO. Why don't you do that so you get every episode? We release one a month, and you can get them as we release them. And if you're interested in what we do, you want to support us financially, go to microbe.tv/contribute. We have a number of ways you can help us out, and we'd be really grateful for that. And of course, if you have questions and comments, send them to TWiEVO@microbe.tv. You can find Nels at selvolution.org. He's also on Twitter as Earlybird, ELEARLY Bird. Thank you, Nels.
Hey, thank you, Vincent. Fun to podcast as always.
Always fun, and I like doing papers where we chat about them. It's kind of neat.
Yeah.
And 2 people is a lot different from 5, which we do on TWiV.
That's right.
We have 2 and 3 and 4 and 5, and they all have a different little dynamic.
Yeah, different energy. Yep. It's good to mix it up.
You get to see insights into papers that you don't see on your own, so that's cool to have 2 people discussing it.
Totally agree.
I'm Vincent Racaniello. You can find me at virology.ws. I'd like to thank the sponsor of this episode, Blue Apron. The music you hear on TWiEVO is performed by Trampled by Turtles. You can find their work at trampledbyturtles.com. You've been listening to This Week in Evolution, the podcast on the biology behind what makes us tick. Thanks for joining us. We'll be back next month. Until then, be curious.